dear student. In last video, we discussed the structure of pollen grain, development of male gametophyte. Today, we will discuss second part of pre-fertilization. In it, we discuss structure of female gametophyte, that's a carpel, structure of ovule, and development of female gametophyte, that's a megasporogenesis. So let's start first. We have already discussed gynoecium. Gynoecium is the female reproductive wall of flower. This gynoecium is made up of carpel. Carpel consists of three parts. That's a stigma, style, ovary, and in ovary there is a presence of ovule. So this indicates the stigma, second style, next ovary, and inside ovary there is a presence of the ovules. This ovule, it may be one or many ovules present in ovary. Next part we see structure of ovule. In most of angiosperm, the ovule is anatropous. That ovule is inverted. The ovule is represent megasporangium. In what's an anatropous ovule? These anatropous ovule are inverted, having micropyle and chaliza in one straight line. So you can see in this image chaliza and the micropyle, it's a one straight line and it's parallel to the funiculus. That form of ovule is called as anatropous ovule. This ovule is represented as the megasporangium. Ovule attached to the wall of ovary by means of fertile tissue, that fertile tissue called as the placenta. We started the structure of ovule. Ovule is differentiated it into two parts. First part its stalk and the second part its body. So let's start the first part, that is the stalk. In stalk, the first one we see, that's a funicle. So you can see this image. This part indicates the funicle. The funicle is the stalk of the ovule by which it is attached to the placenta. This funicle is attached to the placenta. It's a first function. Second, it absorbs the nutrition from the placenta. Second part we see, that's a hilum. This hilum, you can see this point, the hilum, the point of attachment of funicle to the body, means the point of attachment of the stalk to the body, that point of attachment is called as the hilum. The third one, it's a raphe. This raphe in anatropous ovule, the funicle extended beyond the hilum is form of a bridge. It is form of ridge up to the chaliza. It's called raphe. Next, last one in the stalk portion, that is the chaliza. This chaliza, it is the basal portion of the ovule and upper end of the raphe. On upper side of the raphe there is a presence of the chaliza. From chaliza the integuments start arising. This chaliza, it provides the base for the ovule. The second part we see in the structure of ovule, that's a body. Body consists of two parts. First one is integument, Second one is nucellus, and inside the nucellus there is a formation of the embryo sac, or also called as a female gametophyte. Let's start the first one. It's a integument. This integument, you can see this image, integument. The nucellus, inside the integument there is a presence of nucellus, means nucellus is covered by two coat that called as the integument. The outer coat it called as the outer integument, and the inner coat it's called as the inner integument. When nucellus is covered by two integument, when nucellus covered by two integument is called bitegmic. A small opening is left at apex of integument that is called micropyle. So what's a function performed by integument? It gives protection to the nucellus, and after fertilization the integument forms the seed coat. The second part is nucellus. The nucellus, major portion of the body is occupied by nucellus. Nucellus is made up of mass of a parenchymatous cell which is diploid in nature. It is equivalent to the Megasporogenesis. One of cell of nucellus is developed it into the female gametophyte of embryo sac. The third in part we see, that is the embryo sac, which is present inside the nucellus. This embryo sac, it is consist of the 7-cell 8-nucleated structure. Towards the chalices side there is a presence of antipodal cell. Towards the middle side there is a presence of definite nucleus, also known as the secondary nucleus, and towards the micropylar side there is a presence of the egg apparatus. These egg apparatus consist of egg cell and synergids. Next point we start, that's a megasporogenesis. It's a very important part in case of the pre-fertilization changes. The megasporogenesis, it is a process of formation of haploid megaspore from diploid megaspore mother cell. The megaspore mother cell become distinguished in nucellus more or less in the center, but towards the micropylar end of ovule. So let's start the development of female gametophyte in angiosperm. The female gametophyte develops within the nucellus of ovule. A single hypodermal cell is differentiated as archisporial cell. The single hypodermal cell in nucellus, it is differentiated as archisporium cell. A single cell archisporium is differentiated in the nucellus near to the micropylar end. This archisporial cell divide into outer parietal cell and inner sporogenous cell. The inner sporogenous cell act as a megaspore mother cell. This megaspore mother cell, which is diploid in nature, it undergoes meiosis to forms linear tetrad of haploid spore, that is megaspore. Out of these 4 haploid linear tetrad, upper 3 are degenerate and lower 1, which is towards the center of nucellus, which is towards the micropylar side, it remain functional and it act as a female gametophyte. The one megaspore which is towards the center, it is the functional megaspore. This functional megaspore, it undergoes first mitotic division, formation of 2 nucleus at each of pole. It again undergoes second mitosis division and the formation of 2 nucleus at each of pole. Then it again undergoes the third mitosis division, formation of the 4 nucleus at each of pole. 1 nucleus from each of pole migrate towards the center and forms the polar nuclei. The 3 nuclei which is towards the micropylar end constitute the egg apparatus. The egg apparatus consists of the larger central haploid egg cell and the 2 supporting haploid synergid cell. The 3 cell which is towards the chalicer side, it is a function as the antipodal cell. Inside, in egg apparatus, there is a presence of egg and synergids. The synergids shows hair-like a projection called filiform apparatus which guide the pollen tube towards the egg. The 2 haploid polar nuclei of the larger central cell fuse to forms diploid secondary nucleus, or it is also called as the definite nucleus. This method of embryo sac development from a single megaspore is described as a monosporic form of development. In angiosperm, the development of female gametophyte is endosporous, that is within the megaspore. Means the ovule, one of the cell of nucellus, which is act as a sporogenous tissue, it forms functional megaspore, and from a functional megaspore there is a development of embryo sac which is present inside the nucellus, which is the 7-cell 8-nucleated structure. The 3 antipodal cell, in a one cell there is a presence of the 2 nucleus in the secondary polar nucleus and the 3 nucleated condition which is towards the micropylar side. Means the female gametophyte contains 7-cell 8-nucleated structure. Here we completed Megasporogenesis. In Megasporogenesis, structure of ovule we study, structure and development of female gametophyte we study. Thanks for watching my video. We will continue further part in next video. Thank you.